Thank you, Dr. Cha, for introduction, and thank you for this opportunity to share my lab's research for the previous 5 years. Today I will talk about metabolic reprogramming of cancer cells, which help them survive and metastasize. So my lab studies breast cancer and leukemia, and I'll touch upon both cases. So to start broadly, I want to briefly explain about how important metabolism is. So in general, people think that metabolism is just ATP or energy generation, but metabolism is really deeply connected to many diseases. And we often think that metabolic dysfunction is related to obesity or diabetes, but as a metabolism researcher perspective, cancer and aging are very, like close to metabolic dysfunctions. And today, in this, we focus on cancer. So metabolism in cancer cells plays multiple roles, not only for energy production, but also biomass generation, stress resistance, and adaptation to new environments. Works at the interface of metabolism and biosensing. So as Dr. Cha introduced, so my background is electrical engineering. So my labs continue to develop biosensors, but two-thirds of my labs work on metabolism. So on the metabolism side, we are studying how cancer cells change their metabolism for their survival and metastasis. And we are also developing dietary interventions to enhance cancer therapy. And also, we are recently started immune cell metabolic engineering to make it more robust CAR T cells. So I'm gonna focus on the first part of this research topics. So classic concept in cancer metabolism is Warburg effect or aerobic glycolysis. About 100 years ago, a German chemist, Otto Warburg, he actually found that many cancer cells at that time metabolize glucose through the aerobic glycolysis. As a background, so normal cells or normal tissues, they consume most of glucose through the mitochondria to generate ATP. And only in the absence of oxygen, they consume glucose through the anaerobic glycolysis to generate lactate. But Otto Warburg and many scientists at that time, about 100 years ago, found that cancer cells always consume glucose through this glycolysis, even in the presence of oxygen. And at that time, they all agree that cancer cells have a defect in the mitochondria, so they cannot use mitochondrial metabolism, even though there are plenty of oxygens. But actually, as time goes and goes, we people have found that most cancer cells have intact and functional mitochondria, but still they depend on aerobic glycolysis. So I'm gonna maybe frame glycolysis as a more than ATP producing pathway. This is one of the uh, well accepted hypothesis why cancer cells depend on aerobic glycolysis. So glucose through the multiple glycolytic steps make a lactate, a pyruvate, and either pyruvate is metabolized through the mitochondria to generate ATP in the TCA cycle, or pyruvate is metabolized to lactate and secreted out is aerobic glycolysis. But if you see multiple enzymes throughout the glycolysis, they are self-regulatory. So if the cells consume glucose and generate enough ATP, so actually they kind of slow down the glycolysis, maybe spare the glucose for other cells. So they are kind of these self-feedback loops make the cells do not consume glucose that much, kind of like in the right amount. They're only supposed to consume right amount. The problem is that in normal cells, that's fine, but cancer cells, they have to take up like precursor or carbons from glucose for multiple biomass synthetic pathways. If they rely on this mitochondrial metabolism and generate so many ATP molecules, the pathways are going to be slowed down and they cannot provide multiple carbons to biosynthetic pathways. But if the cancer cells rely on this aerobic glycolysis, ATP generation is not that efficient, so that these pathways are not being slowed down, so that they can consume glucose rapidly. At the same time, they can provide many carbon sources to multiple pathways. That's why people think that cancer cells rely on aerobic glycolysis for their proliferation, even in the presence of oxygen. So today I'll discuss more about serine and glycine metabolism. So serine and glycine are very important but non-essential amino acids. So non-essential amino acids versus essential amino acid, it's not divided by their functions. All the amino acids are important, but the reason why they're called non-essential amino acids is basically cells can generate serine and glycine endogenously, like through this serine glycine biosynthesis pathway. But anyway, the serine and glycine are abundant in multiple like types of food, and overall they have benefits in our longevity or good sleep or the reduced risk of heart disease. So in general, they're good metabolites. But cancer cells, their roles are more than that. So as I mentioned, serine and glycine are important amino acids because, especially for cancer cells, serine and glycine are the precursors of DNA, RNA synthesis. Also, they can be used for lipid synthesis as well, which means that as the cancer cells proliferate fast, the needs for serine and glycine are huge. Also, for cancer cells to manage oxidative stress, serine and glycines are used as a precursor for oxidative stress management. So overall, serine and glycine are very critical metabolites, kind of pseudo-essential metabolites for cancer cells. And over the last 20 years or 10 years, maybe 10 years, people have found that multiple types of cancer cells are dependent on SSP, serine synthesis pathway, which generates this serine and glycine endogenously, or the first enzyme, PHDH, in this pathway. People have found that many cancer types depend on this pathway or enzymes. So for example, triple negative breast cancer cells, they analyze which metabolic pathways are highly upregulated in aggressive TMBC versus indolent TMBC. PHDH enzyme or SSP pathways are one of the most highly upregulated in aggressive TMBCs. And also, I found that acute myeloid leukemia, AML, uh, the patient with higher expression of PHDH enzyme, they have worse prognosis. Okay, so I briefly explained serine and glycine metabolism in cancer, and I'll go back, but let's move on about metastasis. Everyone knows that metastasis accounts for the majority of cancer-related deaths, and especially breast cancer. If the localized disease has a much better prognosis, like over 90% of survival rate. But once the breast cancer metastasizes to liver or lung or bone, the survival rate is very small. But actually, metastasis is also very fatal or very lethal for cancer cells.
퍼지는 순간 워싱턴에 어떤 지진이 일어날지
Uh,
최근
okay. So again, metastasis is fatal for cancer patients, but also it's very fatal for cells because it has been shown that millions of cancer cells are shed per every day per gram of tumor. So even before primary tumor is diagnosed, millions of cancer cells are secreted out. But among many cancer cells that are secreted in circulation, very tiny fraction of cancer cells survive, and form metastasis. Because the cancer cells, they have to endure lots of stress conditions to get into other organs. And then once they land in other organs, they have to fight in other cells in that organs. So in the throughout process, the cancer cells face like detachment stress, shear stress, immune attack, oxidative stress, and then like metabolic competitions. So there are multiple challenges are there. And especially as an metabolism researcher, I'm more focuses on two topics: oxidative stress during the circulation and metabolic competitions once they landed. So these two metabolic stresses are my lab's major uh, research interests. And today I'm going to more focus on the oxidative stress parts. So in the breast cancer cells or solid cancer cells, whenever they metastasize, previously people thought that the single cells are detached from the primary tumor and migrate through the circulation. So the E-cadherin, it's a cell, like a cell-to-cell junction molecule as a glue. People initially or previously thought that this E-cadherin is a metastasis suppressor because it makes the cancer cells like bind each other, and the loss of E-cadherin make the cells migrate from the primary tumors, and also it induces epithelial to mesenchymal transitions (EMT). So traditionally, ECAD has been known as a metastasis suppressor. But recently, people have found that actually at least a subtype of cancers, ECAD can work as a metastasis promoter because, um, of course, single cells can be detached and then circulate through the blood vessels and land it into the distant organs. But people have found that cancer cells actually form a cluster for metastasis. And this cluster, the circulating tumor clusters, has way higher potency for metastasis. And these clusters maintain E-cadherin between cell-to-cell junctions, which help them endure stress throughout this journey. And once it lay landed, still this E-cadherin junctions or clusters help them to proliferate fast. So these are two major studies recently published and showing that E-cadherin help the breast cancer cells proliferate and metastasize to other organs. Okay, so E-cadherin can help the metastasis, but also at the same time, metastasis is a very the metabolic stress, metastasis is a full of stress journeys. So my question was how ECAD and metabolism are connected. So to address this question, so we make uh, the two clones, ECAD high expression versus ECAD low expression from the same isogenic breast cancer cells. And then once we make these two clones, we analyze metabolic pathways. And surprisingly, one of the most highly upregulated pathways, serine and glycine serine threonine metabolism, which leads to this serine synthesis pathway. So if you analyze multiple proteomics data between ECAD high, ECAD low cancer cells, multiple enzymes in this SSP are highly upregulated. So we confirmed that with the Western blot. So we tested four breast cancer cell lines. These are intrinsically ECAD levels are low. So when we overexpress ECAD expression, or when we overexpress ECAD in these two cancer cell lines, we saw the increase of PHDH, PSET1, SSP metabolic enzymes are upregulated. And these two cell lines, their intrinsic level of ECAD is high. So in this case, we knock down ECAD and the PHDH and P7 expressions levels also decreased in this knockdown of the cancer cell clones. And also we confirm that this protein level decrease is also consistent with transcription level decreases. ECAD level suppression correlate with PHDH or PSET1 expression decrease. Again, the PHDH, PSET1, there are two enzymes in SSP. So we confirm this metabolic enzyme expression is changed. So we now tested metabolism. So we use isotope tracing analysis with a fully labeled glucose. So in general, most of the carbons are 12C, so 6 positron, 6 neutron. But if you feed the cancer cell with fully labeled 13C glucose, basically you can make it artificially a 13C labeled glucose. And then once you culture the cancer cell with this 13C labeled glucose or any metabolites, you can collect the cells at this given time. And you can, if you analyze which metabolites are labeled with 13C, you can calculate metabolic flux in different pathways. So we feed the cancer cell with fully labeled 13C fully labeled glucose, and we found that ECAD overexpressed cancer cells or ECAD high expression of breast cancer cells have higher enrichment of serine and glycine from glucose, which means that this metabolic flux is higher in ECAD high breast cancer cells compared to control. And also, we tested other extra metabolite analysis. And interestingly, their consumption of glucose are similar between the two clones, ECAD high and low, with two cell lines. But the ECAD low breast cancer clones, they are more dependent on aerobic glycolysis. So which means that ECAD high breast cancer cells, they have higher metabolic flux through the SSP. Also, they have a high metabolic flux through the mitochondrial metabolism. So we are kind of sure that, okay, so ECAD high expressed breast cancer cells have high expression of PHDH or PSET1, and also they have a higher metabolic flux through SSP. So then we ask the question, okay, if we knock down SSP enzymes, can we inhibit their proliferation? Because serine and glycine are a precursor for DNA RNA synthesis. And interestingly, so when these two breast cancer cell lines, ECAD low clones, they barely respond to PHDH inhibition or depletion by siRNA or PHDH inhibitor treatment, but only ECAD high clones respond to this PHDH modulation. And interestingly, once we suppress PHDH or deplete the expression level, their growth rate is similar to ECAD low breast cancer cells. And their expression of PHDH is very low compared to these high clones. Basically, the PHDH expression helped the breast cancers, ECAD breast cancer cell proliferate fast. And by suppressing them or by inhibiting them, their normalization benefit, their proliferation advantage is normalized. So we're trying to understand what makes the cancer cells grow fast in ECAD high breast cancer cells versus ECAD low. And okay, so when you knock down PHDH, growth rate goes down. And when we add nucleotides, again, as I mentioned before, the SSP provide serine and glycine, which are precursor for DNA, RNA. So when we add nucleotides in the culture media, we were able to rescue their growth. And also, but it's not fully grows. So we add also a dimethyl alpha Kg, which is cell permeable alpha Kg metabolite, because the serine synthesis pathway also generates alpha Kg metabolites. When we add it, we also can rescue some, but not even we combine all, we're not able to rescue the effect of PHDH inhibition in ECAD high breast cancer cells. It's similar, but still not there. And it's possible that maybe lipid metabolism also affected, some other pathways might be affected with the PHDH inhibition. So we are now working on it now. So again, the serine and glycans are DNA, RNA, lipid synthesis. So if we just add DNA, RNA, nucleotides, precursors, it might not be enough to rescue the cell proliferation. Okay, so in vitro case, we tested metabolic enzyme expressions, their metabolite analysis, metabolic flux analysis, and their effect on growth in vitro. So we make the in vivo models. So it's also topic on mouse model. So on one side, breast mammary fat pad gland, we injected ECAD high breast cancer cells, and the other side, we injected ECAD low breast cancer cells, and we monitor tumor growth. And as I showed you before, you get high breast cancer cell growth about twice faster, and in vivo case, the growth rate was much different. So the red, black, they are like the control, the vehicle treatment. So this one versus this one. So you get high breast cancer cells grow significantly fast in vivo compared to the in vitro. And when we treat the mice with Ph.D inhibitor, here the ECAD negatives, ECAD low breast tumors, we barely saw tumor growth, so we didn't see much of therapeutic benefits. But ECAD high breast tumors, we saw the significant decrease of tumor growth. So this is the primary tumor growth. So we are trying to ask the question, so really this SSP or serine synthesis pathway help the cancer cells reduce the oxidative stress. So we treat the cells with hydrogen peroxide, basically that acutely induces oxidative stress, compared to like before and after. So definitely after treatment of H2O2, you can increase the reactive oxygen species or oxidative stress. But as you see here, compared to the ECAD low breast cancer cells, the ECAD high breast cancer cells ROS increase is much lower. Uh, and also this cancer cell line also ECAD high clones are very, the increase of ROS is much lower than ECAD low clones. And when you inhibit the PHDH on these ECAD high clones, the increase of ROS is more than the control groups, and both cell lines are consistent. And then we analyze why there is difference in ROS levels, and we realize that the ROS reducing capacities are different. So GSH is glutathione. So basically in the cells, when they reduce the oxidative stress, they use glutathione as a ROS reducing agent. The ratio of GSH over GSSG is a matrix or as a indicates capacity of cancer cells like reduce oxidative stress. Again, ECAD high clones have much higher capacity than ECAD low clones, and the capacity decreases as you suppress the PHDH enzymes. And the next step or last step, we tested micrometastasis models. So previous in vivo models, we basically inject the breast cancer cells to monitor tumor growth. But in this case, we just inject the breast cancer cell through the IV, and then after 48 hours, we collect a lung localization. It's basically micrometastasis step because we are more interested in this circulation and the initial step of colonization of in the whole metastasis. And we have three groups: vehicle treatment, PHDH inhibitor treatment, and PHDH inhibitor with NAT, which is ROS scavenger. Again, if you suppress the PHDH, the ROS level slightly goes up. So in compared to the vehicle groups, the PHDH inhibitor treatment breast cancer cell, they have higher ROS levels. And when you treat them with NAC, then you can reduce the ROS even with the PHDH inhibitor treatment. After this overnight treatment, we inject them into the IV, intravenous venous injection. And we found that breast cancer cells were pre-treated with the NCT503, which is PHDH inhibitor. The number of colonization or initial colonization was significantly decreased. But when we treat the cancer cell PHD inhibitor with the ROS scavenger net, actually you could see the increase of metastasis, which means that during this micrometastasis step, cancer cells with higher ROS cannot survive and cannot make the localization. But if the cancer cells are treated with antioxidant or have lower ROS, they can maintain or they can endure the stress through the circulation and they can land it in different organs. Again, in this part, we studied how cancer cells endure metabolic stress through this circulation. And then this is our model. So two breast cancer cell, one is ECAD high versus ECAD expression low. ECAD high expression cells, they have higher metabolic pathway, serine synthesis pathway, which help the cancer cells reduce ROS stress because they can generate a higher level of antioxidants. Also, it helps to generate nucleotides, DNA or RNAs, so that which help them to proliferate faster. So this is the ECAD metabolism metastasis link, and we are now studying more in depth the detailed mechanisms. So this is part one on this today's talk. So part two is more about leukemia. So acute myeloid leukemia is not a common disease, but it's a very small disease. About more than 50% of patients experience the disease relapse, and once it relapses, the 5-year survival rate is less than 35%. And about 10 years ago, FDA approved drug venetoclax was a blockbuster at the time, but still not much effective in about half of the AML patients. And even with the patient who responded well at the first, but they often develop resistance. So in the field of leukemia, the enhancing the treatment efficacy is really important. And surprisingly, this cancer cell paper have pointed out that the bone marrow of leukemic patients have a very high level of fructose. So bone marrow is the word in the middle of the bone, and in which the AML cells are on the reside and kind of proliferate there. Because leukemia is from the hematopoietic stem cells, so their major tumor microenvironment is bone marrow. And in normal people, the fructose level should be less than 1 millimolar. It should be less than zero point 0.2 millimolar. But somehow, still, the mechanism is not known. The bone marrow in leukemic patients have a very high expression of fructose. So what's the fructose? So basically, you might hear many times about high fructose corn syrup in Western diets. Maple syrup or sugar is like 9% sucrose. And then this one sucrose molecule is a combination of one glucose and one fructose. So if you eat the sucrose or sugar, it goes into your small intestine, and the enzyme in the small intestine break down so that glucose and fructose are metabolized in separate pathways. And if you see how fructose is metabolized in molecular levels, actually it's very interesting. So major fructose metabolism organs are intestine and liver, and this ketohexokinase enzyme metabolizes fructose through the fructose 1 phosphate. And either it can go and join the glycolysis, or it goes to metabolize to other pathways. And other organs, they don't have ketohexokinase, so most of the fructose is metabolized hexokinase. But hexokinase mostly focused on like hexokinase also metabolizes glucose here. So whenever the glucose is present, hexokinase are busy for handling glucose. So in normal cases, cancer cells, like other organs, other than intestine, liver, they don't metabolize fructose. So, because people have pointed out that in the tumor microenvironment, including bone marrow, people have pointed out that the glucose levels are very limited because rapid growth of cancer cells leads to like rapid consumption of glucose, so that in the tumor microenvironment, normally glucose levels are very little, and then people have suggested that in leukemic AML, leukemic cells may metabolize fructose in the tumor microenvironment or bone marrow. So we tested. I cultured the four different leukemic cells in glucose media, so regular media, or remove glucose only fructose media, and then no sugar media. And of course, there's no sugar. Cells don't grow at all, as this green line shows. And surprisingly, and among the four of leukemic cell lines, their growth rate is very similar between glucose media, fructose media. Again, in the fructose media, there's no glucose. And one of the cell lines actually grew faster when they are fed with fructose. And we do more metabolism analysis. Surprisingly, even though they grow a similar rate or a faster rate, the consumption of fructose is very little compared to the glucose. So basically, compared to the glucose microenvironment, in the fructose microenvironment, the consumption of fructose is very little compared to glucose, which means that overall metabolic flux might be suppressed or reduced. Instead, leukemic cells upregulate glutamine metabolism to compensate the slow metabolic rate of fructose. But interestingly, we found that when the cancer cells are cultured in fructose microenvironment, the glycine peaks are highly upregulated. And when we do multiple metabolic screening, glycine and serine levels are much more elevated in the fructose culture conditions compared to the glucose culture condition. Again, we also these are metabolite in the media. So we studied intracellular levels of metabolites using this isotope tracing analysis. And as we when we grow the leukemic cells, a certain C fully labeled glucose or certain C fully labeled fructose, we saw serine and glycine levels are not only total levels, but also the certain C enrichment levels are much more higher in fructose culture conditions. Okay, again, in the culture media, serine and glycine levels are higher, which means that cancer cells make the serine glycine and secrete out. And also inside the cells, the serine glycine levels are also much higher. So we thought that, okay, compared to the glucose condition, leukemic cells metabolize fructose. Overall, fructose uptake rate is much slower than glucose uptake rate, but the metabolic flux through the SSP, serine synthesis pathway, might be higher. So we knock down PHDH, again, the first enzyme in this pathway. When we knock down PHDH, the glycine peaks are gone, which means that all the labeled glycine or serines are generated from the SSP or serine synthesis pathway. And when we compare the proliferation, when we knock down PHDH, the growth rate decreased also in glucose conditions, but in the fructose conditions, the growth rate decrease is more significant, which means that leukemic cells rely on this serine synthesis pathway or PHDH for their proliferation under the fructose-rich conditions. Importantly, normal CD34 positive cells, which are like hematopoietic stem and progenitor cells, which are normal counterparts of leukemic cells, they do not increase metabolic flux through the SSP in fructose-rich condition. Again, leukemic cells, they showed significantly higher level of serine and glycine in fructose media or fructose culture condition, but normal CD34 positive cells, the levels are overall similar, and also the enrichment level of serine and glycine from 13C labeled glucose is also much changed, which means that normal hematopoietic stem cell progenitor cells uh, do not increase this flux to the SSP. And again, MOM13 is AML cell line. They are significantly dependent on Ph.D. GDH or SSP because as you see here, when they are treated with this inhibitor, the growth rate was significantly decreased under the fructose condition. But normal hematopoietic stem and progenitor cells, which are counterparts of these malignant cells, their growth rate was barely affected with this PHDH enzyme inhibitor. Which is interesting because normal cells do not respond to the drug, but leukemic cells respond, especially in the fructose condition, respond more, which is very important because I mentioned before, leukemic tumor microenvironment, the fructose levels significantly higher and the glucose level is low, which means that leukemic cells are likely to expose to fructose condition, rich condition, which means that leukemic cells or in the bone marrow, they are very dependent or they are very sensitive to PHDH inhibitor. And we also tested AML patient cells in glucose fructose condition. And when we treat the PHDH inhibitor, the annexin-5, which is apoptosis level, it was much significantly increased when we treat them with drug compared to the glucose condition. Of course, glucose conditions, apoptosis was increased with the drug, but more significantly in fructose conditions. Okay, so we kind of understand, okay, leukemic cells, they increase the metabolic flux through the SSP in the fructose condition. And then what's the benefit? Or what's the benefit and what modulates this metabolic flux? And we found that NAD plus NADH, which is a cofactor for this enzyme PHDGH, were significantly higher in fructose conditions. So NAD plus levels slightly higher in fructose culture condition, but NADH levels are significantly lower compared to the glucose condition, which means that the PHDH enzyme, which uses NAD plus as a cofactors, the reaction rate could be much higher in fructose-rich conditions. And so we play around the ratio. So if you culture the cancer cells with supplemented lactate, you can actually decrease the ratio because once the cells take up lactate, is metabolized by lactate dehydrogenase, make a pyruvate, but at the same time they consume NAD+. So when you feed the cancer cells with lactate or any cells, it takes up and then consume NAD plus so that the ratio goes down because NAD plus level goes down, NADH level goes up. And as the ratio goes down, the glycine levels are significantly dropped. Again, this is acetylserine synthesis pathway, and this PHDH enzyme uses NAD plus as a cofactor. Lactate supplementation decreases the ratio, which decreases the metabolic flux. On the other hand, if you feed a cancer cell with pyruvate, the cancer cells take up pyruvate and metabolize through the same enzyme LDH, but in this case, they consume NADH to generate NAD+. So as you see here, the ratio can go up with the pyruvate supplementation. Again, NAD plus as a cofactor of PHDH. So when you feed the cancer cell with the pyruvate, the ratio goes up, more metabolic flux, and you can see higher glycine levels in the media in the glucose culture conditions. So NAD plus levels or NAD plus NADH ratios determines the SSP metabolic flux, and which indicates that higher ratio indicates higher metabolic flux in fructose-rich conditions. And also, I'm not gonna explain more, but one of the metabolites that can generate alpha Kg, which can go into the mitochondria or help them anaphylaxis. So lastly, we tested this in vitro metabolic reprogramming with the AML leukemia mouse models. And once we make the AML mouse model, we feed the fully labeled glucose or fructose or Ph.D. inhibitor with the fructose and then we saw the glycine levels, how much are labeled. And when we feed the or injected the leukemia mouse model with fully labeled fructose, we saw more glycine enrichment in the bone marrow compared to the glucose, fully labeled glucose injection. But when we inhibit the PHDH with the drug, we saw the decrease of glycine enrichment in bone marrow of leukemic cells. Although the p-values are not like less than 0.05, but we believe that there's a trend because it's not easy to deliver the metabolized drug through the bone marrow in the mouse models. Lastly, we tested the glucose levels and fructose levels in the bone marrow. And then leukemic mice, again, the glucose level is much lower than healthy mice because I guess the leukemic cells take all the glucose, so that decrease the glucose levels, but actually their fructose levels are higher. And the fructose levels are even higher than glucose levels in the bone marrow of leukemic mice, which is consistent to the human data. Again, we still don't know why the bone marrow they have higher fructose levels, but it's possible that leukemic cells are facing the glucose depleted condition and they start consuming fructose, which induces increase of metabolic pathway through the SSP, which means that if you target this PHDH in the tumor microenvironment, you might be able to increase the therapeutic efficacy of this dismal disease. As a wrap up, so these leukemic cells proliferate well in fructose-rich conditions, and then this flux is increased with the higher ratio, which facilitates the alpha K generation, and then overall provides metabolic advantages for proliferation. So with that, I can stop now. Again, my lab at Hopkins works on metabolism and biosensors. And today, I briefly discuss about what we do about the cancer metabolism and mainly focus on serine and glycine metabolism, which are, again, I can go back to this slide. They are non-essential amino acid, but for cancer cells, it plays critical roles in DNA RNA synthesis, lipid synthesis, and oxidative stress management. So these metabolic enzymes or overall these pathways has been discussed many times in cancers, and my lab is also very interested in this part. So I can stop now, and thank you for your interest and thank you for your patience.